Hello.
Bradley.
How are you?
Good, good. Last week, actually, Mehul was there in the meeting, so I didn't know that because I somehow we actually pretty much discussed, and also I have given the same update to him so that So
Great.
we discussed, but I didn't have the result at the time. But when he was there, I gave the update. So probably I can give quick updates on what I have done.
Yeah.
And there were pretty much large changes what happened last week and all. So in the last meeting, what I have discussed is from the beginning of what I have done is initially I started off with understanding the data set where we have like the XYZ coordinates of the C elegans, each time point for the during the embryogenesis, and also we have the lineage information, how it's evolving over the period of time. So what I wanted to understand in this is more specifically apply a hypergraph on top of it and see how this hyper edges the edge that actually contributes to bring a certain set of hypernodes together, and what is the underneath information it resides, and which certain type of cells lead to anastomosis. So that's my major understanding. That I wanted to understand practically using a data set. So where I have in the first couple of weeks, I started with implementing the basics of implementing the first I've decided the workflow and the dependencies and the libraries. So I have figured it out, and later I implemented a few of the neural networks. And I thought there is a pipeline that actually can help in order to integrate it. But in the week 5, the previous week, I was able to understand that the pipeline that I thought was not helping. So I had to change and tweak a bit in the networks. So what I have tweaked is probably I'll share it now. So yeah, so these are basic dependencies and networks. I have taken this cell birth and position dataset because the temporal ones is quite long. So this could be short, and I will be able to acquire the information from this. And today what I've decided is I'll probably run the results and show it like how this network is able to understand. So we have this the cell parent information and the XYZ coordinates, and also the the daughter 1 and daughter 2, how the parent has been split into that and the given time point. And from this, I was able to get some node level features for the for a hypergraph. So I say at each node, which means in a graph we have a node, and in this node, what are the information it has? So we have the time, and we have the XYZ coordinates. This is what it actually has, and the directed lineage information. The parents, the lineage information is also resides into the edge. Saying that this the lineage, how it traverses exactly. So we have the information that actually it resides into the the edge, saying that the hyper edge says that that what kind of grouping that we are doing. So it's basically based on the the relationship between the lineage tree that it has traveled from, like parent to the daughter, and from that specific daughter splits into like multiple daughters. So it's like that. So first generation, second generation, and further generations. So I was able to acquire 1203 nodes and 1212 lineage edges that we were able to found. So we have built a hypergraph where we have different all these features that have been acquired. So in order to have like an hyper edge, so we were using D-HNN with a certain radius, so that we will be able to acquire the neighborhood embeddings, so that spatial information is already sorted. Because in the edge we are anyway we are acquiring the information of the lineage. So here we are trying to acquire the information for the the spatial, I mean like XYZ locations. And what in the next step, what we have done is, so we will be able to acquire all these matrices, where I'll say incidence matrix is basically like adjacency matrix for an hypergraph. And so we have this information of lineage information, and we have the information of the spatial locations, and we have the birth time positions, and then we will make a hypergraph. Here I have specifically made it alive because I also wanted to add a further next set and features which we discussed in like previous meetings, that we also have the cell depth or birth information, so the fate can be predicted. So I thought, okay, if you're going to predict the fate, then probably this information could be helpful in the future run. So I just added that. And after that, we are able to acquire. I changed this hypergraph neural network a bit. So where I have changed and why do I have to change is the major concern. So last time what I have done is I tried using lineage information and also the spatial information. Based on the lineage and the spatial information, we were able to design an hybrid type of model. So what it is not able to do is, so we do not have a, it has basically a temporal line where it says that, at one time point, it has a certain characteristics. At time point two, it has certain characteristics. So the network that I have designed previously was not able to understand this, the temporal relationship. In order to make it understand the temporal relationship, so I added one more component here, so which is the graded recurrent unit. Or we can use LSTM or we can use transformers, but as of now, I started with gated recurrent units, so which actually helps to understand it how we're going to progress over the period of time. And so once we are done with the neural network, we have like graph convolution layers, and we have like temporal connections, and these embeddings will be helpful for us to build the time snapshots. So what I have decided in the previously in order to train it in a certain way, but this time I had a change in perspective. So when I discussed with Mehul, he said that unsupervised fashion is not required as of now. You have a proper label data, so probably you can work on this label data. So I was like, yes, I think, I mean, like, I thought it's a better way to do it. So I didn't do the, I applied the self-supervision. We were able to not get any results because it's not able to understand well from the data. So I've tried like proper supervised approach, where what I have done is here, the critical modification I have done is like, I have two losses. One actually tries to give us, the loss, I mean, like the change from the prediction and the ground truth is basically mean square error. So what is the next it's trying to predict, and what it actually has, it tries to bring them together, and also it tries to do the a binary cross entropy between the incidence matrix. So using when we try to compress this incidence matrix, it actually the network topology does not lose its topology. So the topology remains well using this this loss, because we just try to minimize the the distance between 2 points is not sufficient. We have to make sure that the structure is not lost. So we added extra top of loss, which is like incidence BC. It tries to minimize the cross entropy loss between the incidence matrices predicted and like the actual ones or the ground truth. So when I trained this, I was able to get it's converged to a certain point, and once it's converged, I wanted to understand that what these hyper edges. I mean, like the main motive of this is what does the information in our hyper edge carry. So how are these embeddings? So when I plot the TSNA, I was able to see this plot. So I'll probably run this so that you can have a quick intuition. So helper function. And this is broad times information. This is our neural network, and this is our alive or not information. Yeah. So snapshots are very, this is our last function. This will take a couple of time, not much time, but little time. So once it's done, we can probably plot them. So I saved the plot here so that it looks similar to this. I mean, like, you know, the embeddings during the division and the hypergraph, whatever hypergraph that we built, was able to bring certain set of cells together. So these cells, I haven't annotated them yet, but so this is AB. So we'll have all the annotations together. So once we will able to annotate together, then we will be able to see that what is our hypergraph learning. So is it learning that, you know, brings all these cells together? What does it mean? So that interpretation probably I'll try to do it next week, and I'll bring that report or kind of PPT so that it's more elucidated. So what I'm trying to understand here is, you know, during the training of this model using a hypergraph, we were able to see that the hyperedges tries to learn certain set of cells being together. There are certain lineage structures that are coming together. Do they really form a certain type of characteristics? I mean, like, let's say, you know, the interneuron come together, the motor neuron come together, or I mean, like, you know, locomotory cells come together. I'm not really sure about what comes together, but I have to, you know, experimentally observe what are these getting together. So these cell embeddings will actually help us to more understand, you know, what is the topology, the underneath topology of, like, you know, this growth happens. So this is TSNA. We can also use UMAP. UMAP actually gives us more intuition about, like, the network topology. I actually added the function here too. So yeah. So this is the, you know, there are certain cells that are like coming together. Yeah. So yeah. So that's the main thing. So here TSNA we observe it's a different way, and this is different way, but at the end of the day, what we try to interpret from, like, TSNA is the distances here are like does not mean anything in TSNA, and in TSNA, you know, the groups that have been formed there, when we really annotate and give them proper colors, then we will be able to interpret more in better fashion. So I mean, so this is the update that I have done for this week, and probably in the next couple of weeks, I'll try to understand more in detail, interpret them, and apply, you know, a different type of neural network where we can get more interpretable features, or say that, you know, how well it's trying to bring new insights, or probably we can say that, you know, does the network topology really matters, or if we change the neural network, are we seeing the same cells forming them together? So is it what the neural network learns, or what it actually happens? So I mean, like, I have few questions, and probably I'll formulate them in a more better manner, come with some experiments, and then probably we can discuss. So I think this is what I have done for the last week, and I have just shown the code-wise update, but I think it would be more better if I give like a formal type of presentation, which I'll probably do next week.
Okay.
In this Friday. So
sounds good.
So any suggestions?
Yeah. Well, I like the sort of bringing this all together in a visualization, and then showing, you know, kind of how things are coming together in alternate structures, the T-SNE maps and so forth. I mean, I don't know how those would be. I don't know if they're also related, sort of as a lineage tree or like a reorganization of the lineage tree. But you know, you could say like that they're grouped in some way. I don't know if, well, do you know for a fact, or do you know that if they're basically the same as a lineage tree, or are they bringing together cells from different parts of the lineage tree?
Right. That I need to understand by labeling them and then extract the information from them.
Oh, okay. You haven't labeled them yet.
Yes.
Oh, okay.
Yeah, I have labeled them, but while plotting, what it happens, it overlaps a lot. So when it overlaps, the visualization does not look clean. So I had to think of like a more cleaner visualization where, you know, when I point it out on top of that, then I should be able to see that, okay, it started from this, or this belongs to something, and so I can.
So what do you?
So
I
any
mean,
thoughts?
yeah. So you talked about kind of the whole point of this was to show how the edges inform different parts, different like hypergraph constituents. So a hypernode would be like have a certain number of cells. Depending on the edges, they have different relationships. So
Right.
yeah, so that's how you kind of got to the T-SNE maps, or I mean,
Yeah.
thinking
So
about that.
what I
Yeah.
have done is to extract the TSNA. So we have hyper edges and hyper nodes, and these hyper edges basically form across the nodes. So what I thought is, okay, I made these hyper edges learnable. Learnable in the sense they can change across, time, trying to learn that which hypernode should come inside the hyper edge. So they are not like static per se that only these have to come to the hyper edge. I mean, like suppose I say that there might be a cell which is proximately closer, but they are different. But the lineage information is different. It tries to bring both the lineage and balances out between lineage and also the spatial proximity, and it tries to learn that it brings together the a certain type of cells which it thinks that it is closer to the ground truth what we have. So during that process, the hyper edge tries to learn to bring certain type of cells together. What I wanted to I plotted is the weights which the hyper edge has acquired. Hyper edge in the sense the edge which is actually responsible to bring out the cell-wise relationship. I plotted the weights of that hyper edge. Then I thought I'd be able to bring out the cells which are actually more relevant, or it's more of which actually bring the how should I say this? So it tries to bring out the more so cells which are actually more relevant, and it tries to understand which cells actually form together, and this hyper edge I was able to plot it. The embeddings of the hyper edge.
Okay. Yeah, that looks great. So you'll just keep working on this kind of. You said that your original pipeline didn't really work very well. So
original pipeline, what I thought was to apply the unsupervised manner. Unsupervised manner in the sense I thought, okay, without giving the labels, I should understand just by the data. So which was not really working. I actually needed, what is the actual ground truth of the next time step prediction. Without the next time step prediction, I was unable to get any results out of it. So there was no temporal relationship to find out that it starts from like a 0th time step to like 100th time step. There was no relationship. I mean, like there was no network which actually tries to understand the space, the temporal relationship.
yeah.
So in order to understand that, I have to embed one more the GRU or LSTM or transformer block where it actually has the information that at time point 0 it is here, and time point 1 it is here, and the incidence matrix is also like a very good approach. So when I discussed with Mehul, he was like, okay, this actually preserves the network topology. Then you're not breaking out what actually the graph it actually has. So the relationships are not lost, while trying to model this. So that was the key insights which I have observed this way.
Yeah.
So that's from my side.
Yeah, that's great.
I'll probably bring more intuitive visualizations and intuitive books this week.
Yeah. Well, that's great. Yeah, thanks for the update.
Okay, Bradley.
Yeah.
Yeah, I'd like to show you a confusion I'm getting out of the literature.
Okay.
And see if you can resolve it.
All right.
Let me share my screen.
All right.
Okay. Start screen sharing. Share. Okay, now. Oops. No, that's not it. Wrong thing. Bye. I'm having trouble here because the Jitsi covers up my dock.
Oh, you can hide it. Yeah.
I can hide? Oh,
Yeah, it
hide
says hide
this.
there. Yeah.
Oh, hide it.
There you go.
Oh, okay.
Yeah,
There we
yeah.
go. Thank
Yeah.
you. Okay, now. And this is a table from this paper. Okay. But Podbilewicz worked with white. Okay, so this is goes back. His pedigree goes back to the original work here, I guess. Okay, now this is a table, and you'll notice that it's a table that claims that there are 44 syncytia. Okay.
Yeah.
Okay, and this is the number of nuclei in each syncytium.
Okay.
Okay, the confusion I'm getting is the cells are named here, but I can't tell if that's the name of the cell from which the syncytium came or the name of the syncytium cell.
Right. Right.
Okay, and I've gone through the paper. I can't figure it out. It gives a bunch of references here, and I just started getting those references, and I'm not sure whether or not they'll help resolve that issue. But the paper just doesn't tell me that question. Are these the cell from which the syncytia came, or is it the name of the syncytial cell? Is there a simple way to figure that out, given the name of a...
Yeah.
If these cells are on the lineage tree, how do you go from a name to locating the point on the lineage tree that corresponds to this cell?
Well, yeah. So these names are the cells from which the syncytium came. So
How do you know that?
Because you can see it in the lineage tree. We have the interactive lineage tree that you
We
can
do.
look at.
Oh,
Yeah,
okay.
and that goes
Can you give it-
post-embryonically as well as embryonically.
Yeah,
So
can you put that?
post-embryonic, yeah.
Okay. Can you put that in the chat?
Yeah, I don't have the link right now, but I can get it later.
Okay. So in other words, the interactive one would allow you to put in a name of a cell and locate it on the lineage tree?
Yes. Yes.
Okay, great. And are these, do you recognize these as being on the lineage tree?
Yeah. Yeah. I'm gonna get to this in a little bit actually.
Okay,
I have some visualizations to show.
Now the next question then is: are these the progenitor? I guess here's an example here: Hype 6 plus Hype 7. We have two progenitor cells for this huge syncytium.
Mm-hmm. Yeah.
Okay, so that's that's an example where we're getting a mix, but it looks like the rest are straightforward.
Yeah.
So this is the only paper I've been able to locate which explicitly says there are 44 syncytial cells.
Right.
Okay, and, I think they're distinguishing the male from the hermaphrodite.
Yeah.
The number of nuclei report differences were males. Okay, so there's a small group of those. Okay, now what I'm suggesting is very simple, a simple form of hypergraph. Those that come from a particular cell could just be shown off the lineage tree as a circle with the number of nuclei in it.
Yeah.
Okay. This would have, if this one could be represented the same way, but it would have two connections to the lineage tree. Okay. Yeah. So I think this gives us all the data from which to construct a hypergraph where we don't. The hypergraph is just a lineage tree connected to a circle with a number of nuclei, and then connected back to the cell from which it came.
Yeah.
The circle is connected to the cell to which it came. Lalith, do you agree with this approach?
Yes, I mean, like, I believe that, if these cells actually, they come from the Syncytium, or Syncytium actually forms these cells, I'm like I'm unable to understand that. But once I'm able to understand that, then probably it means that, cell
Yeah.
which actually is that
I'll get these papers where the reference. Let's see.
Right.
Oh, here's the superscripts are over here. Okay. So I'll get these papers, and I think we should go through them carefully to make sure we understand this. This graph is hard to understand because I am not familiar with the names of individual cells, and I didn't know there was an interactive lineage tree, which is great.
Yeah.
So we could modify the lineage tree easily, it seems to me, simply by representing the syncytial cells by a circle with the number in the circle, which is the number of nuclei, and attached to the cell from which it came, or the cells from which it came.
Yeah.
Okay, good. You've clarified this tremendously for me.
Yeah.
Thank you.
You're welcome.
Okay. Okay, so we've got a lot of work to do to get these references and make sure and get that interactive lineage tree so we can then make the hypergraph. And what's nice about this is we get to make, assuming this data is correct, we get to make the hypergraph for all of the syncytial cells, not just one.
Yeah.
Okay, so we can really make a modified lineage tree which includes syncytial cells, which I haven't seen anywhere. Have you?
No.
Oh, okay. So I think it's a weird thing that's missing from the literature, because there's lots of work on syncytial cells in C. elegans, but nobody seems to have made it to modify the lineage tree to account for that.
Yeah.
Okay.
Yeah.
So it's a large project, but well, actually, it isn't that large, because by reducing the syncytia to a circle with a number of nuclei in it, I think it's easy to add to a lineage tree.
Right.
Okay.
Right.
Okay, that's great.
Right.
Clarify what work lies ahead.
Yeah.
Yes. Okay, please do get us the URL for the interactive lineage tree.
Okay. Yeah.
Okay.
All right.
Okay, that's it. Thanks.
All right.
Okay, Lalith, did you follow all that?
Yes, I was doing for
Okay. Thanks. Yeah, I think this paper is an excellent source as long as one understands the table.
Yes, I mean, like, I was able to understand only that paper.
Okay. Yeah, I didn't understand it when I got it, and I read the paper and it didn't explain it. Okay. Problem with people assuming you know their previous work.
I noticed a lot of the tissue is epithelial tissue.
Oh yeah,
That's
Yeah.
interesting.
we could either color label them the little circle. Probably the best thing to be color label the circles representing the syncytia according to cell type. Okay, that might be a useful addition. Thanks, Susan.
And so I'm going to now show some work that I've been doing on the lineage tree and kind of visualizing some of these relationships going through some of the specific Syncytium that are in the C. elegans lineage tree. So this is the interactive lineage tree here. This is wormweb.org, and you often use this to find cells. So you can search for cells here. So I can search, it starts at P0 and it goes down, and it has the terminal fates labeled. So you have these rectangles with names on them. Those are the cell nomenclature. You have a time scale at the left. You can go down through like the lineage, and you can keep going down. And you have these developmental cells, which are the cells that kind of divide from P0, that divide into the sublineages. You have the founder cells. I'm following down the AB lineage here. I'm following down to some of these cells that start to change their nomenclature. So these AB cells with the suffixes are all part of this AB sublineage. But then we get to something called P10, which is a different type of cell. And if we click on it, it's a post-embryonic blast cell, ventral cord neuron. So you start to get these differentiated cells in the lineage tree. You get things, the other like QR cells, and this is hypodermal 7, which is actually part of one of the Syncytium. So you get that cell. I couldn't search for it, but it shows up. Hypoderm 7 has a lineal name, V5RA. It's hypodermal, or it's hypoderm. It's born at 1100 minutes, which is post-embryonic, and then it's terminally differentiated into this hypoderm, and it's actually then has this additional lineage trajectory where it fuses with other cells in Hype 6 and Hype 7 in that class. So that's how they kind of represent this. It kind of stops at these individual cells for the Syncytium. And then, of course, you can get these cells to fuse, and then there are additional rounds of nuclear division that happen. So that's why you have that discrepancy between the number of input cells and then the number of nuclei, because there are rounds of nuclear division that happen after that. Again, it doesn't show up in the lineage tree. So that's, and then I'm not going to go through, what was that?
Bradley, this is where we would add the Syncytium then.
Yeah, down below this.
Okay.
And so this is during the L1 stage of larval development. So that means that, we have these 4 stages of larval development, L1 through L4. A lot of the precursors to the Syncytium are happening in L1. And then the structure itself, I think, that this contributes to, it forms an L4. And I'll show a graphic of that in a minute. But just to give you an idea of what the cell lineage tree looks like and how you can, there's this nice tool where we can get that information and play around with the visualization here. So this was made in, I'm not sure what this was made in, but this has been around for quite a while. So, yeah.
Is the original code for generating this available?
Yeah, I think Nikhil Bhatla, who runs this, I think he has a GitHub repository for it, but I'm not really sure.
Okay, so we might be able to modify his code to show the Syncytium.
Yeah, wormweb.org is where you can find more apps. I don't know if he has a link to a GitHub repository, but yeah, this is I don't exactly know what this is made in, but anyways,
Okay.
yeah. So that's the interactive lineage tree, but that's of course branching. So you're going from like the.
That's the question. As you said, the time scale's on the left. Is there time data available for the formation of the Syncytium?
Yes, in the literature, there's information on this.
Okay, so we need to do some digging then.
Yeah, well, I'll get to this in a minute. So yeah, this is just the branching relationship. So this is how you go from sort of the developmental lineage tree, which is defined by these letters A, B, P1, that whole nomenclature. Then at some point it switches over to these post-embryonic cells, which have a different nomenclature. And so there's a lot of cell division happening in embryonic development when the worm is in its egg, and then it hatches with maybe about 2/3 of the cells that it needs that are kind of there. Some of them terminally differentiate in the egg, but then some have to wait until after in this larval development to differentiate. And that's what this post-embryonic lineage tree is. So Sulston published 2 papers, John Sulston. In 1977, they published a paper on the lineage tree. And then in 1983, they published a paper on the post-embryonic lineage tree. So they worked out the lineage tree in the egg, in the embryo, and then they published a paper on this post-embryonic lineage tree, which involves things like, syncytia, different structures being finalized, and, seam cells and things like that contribute to morphological changes within larval development. So you end up, you start at L1 with this smaller worm that's moving around. It's missing some things, and then those things kind of form by L4, right before adulthood. Usually it's things to do with reproduction and other types of structures. And there's also a significant amount of environmental plasticity that can happen in the larval developmental stages, which involve remodeling of the phenotype. So you get dour worms, you get starvation, like you can induce starvation, and the worm will basically freeze itself in development, things like that. So there's a lot of stuff going on in this post-embryonic phase.
What is the relationship between White and Sulston?
I'm not really sure. I think they were both at Oxford, but other than that, I'm not sure.
Okay, because people have done a lineage tree for certain academics, and it might help sort things out by knowing the relationship between this guy, the guy who generated this table, and other people he's worked with.
Yeah. Well, I think they were colleagues, yeah, but I don't know.
Okay, yeah.
Yeah.
Yeah. Okay. Okay. The other thing, here, let me put this. I use for family purposes, I use a program called Family Echo. Okay. And what this is it's a tree of families. But what it has, which the lineage tree does not have, is that it allows for marriages.
Right.
Okay, which are in a sense are fusions.
Yeah.
Okay.
Yeah.
So it might be worthwhile looking at Family Echo to redo the tree if we feel that it's worthwhile doing.
Yeah.
Okay. You might want to play with that for your own family. see how it works.
Yeah.
But it allows for each person, it allows you to put a picture in and all sorts of data about them.
Oh, good.
Including contact information. Okay.
Yeah.
So as your life goes on, it can be a rather dynamic thing.
Yeah.
Okay.
Yeah.
I suggest you at least look at it, see if Family Echo is attractive compared to standard lineage trees.
Yeah. Okay.
Yeah, yeah, I did it for our family, and I actually got individuals back to the 1700s.
Wow.
And you do that by looking up obituaries.
Yeah.
Okay.
Yeah.
So by the time it was done, I think I had over 400 people in my family.
So yeah, let me show these. So this is something I've been working on for this. If you go into the literature, there are different types of structures that you can look at, and so this is actually, let me actually grab. I don't know if I can do that. Okay. So this is one such structure. This is a structure where there's a syncytium being formed down here. And you can see that the way that this is kind of laid out is that we're going, and a lot of this is taken from both the literature and from the interactive lineage tree. So we start off with our developmental cells very early on, establishing which developmental lineages they're coming from. So we're starting with this AB sublineage. We're starting with the posterior end of that. We have these left and right divisions. And then we have these three categories, P5P, P6P, and P7P. So the developmental lineage tree, tracing this from 135 minutes with these different developmental cells here. And then we go down, and there's a break in the timeline to 1,340 minutes, that's L1. And during L1, which is this, remember this post-embryonic period, you get these three cells, P5.P, P6.P, and P7.P. And those then go on to form these other precursor cells. We have A, B, C, D. We have this forward and reverse, and then we have E, F, forward and reverse. And these go on to form other cells. Those cells proliferate, and then they start to fuse in L4. So this proliferation takes place in L3, and then at L4 we have this 2,780 minutes. We have this structure forming, and so this is the structure, and it's kind of like there's contributions from each of these cell types, and that's how they're kind of structured. So that's one way I kind of visualized this structure. Then there's this other way of doing this where you have this lineage tree. We have this other, this is where we have our hypodermal cells. And they're kind of forming these, they're coming from the developmental lineage tree, and they end up as these type of precursor cells for a syncytium. So you have your sublineage C, and you go down, you have the anterior and posterior direction. In the anterior direction, you have CA, CAA, and then you get these hypodermal cells, hypodermal 7, more hypodermal 7 over here, and you get these at different time points. So that's what these numbers in parentheses are. You get 4 here from CAAA, 1 from CAAP, 4 from CPAA, and 4 from CPAP at slightly different times. And so those are all kind of in place. And then for B, this is kind of working out more of these hypodermal 7 cells, again from the AB sublineage. So you have a sketch of this, how this sublineage breaks down. You get cell divisions in developmental cells, and you get these hypodermal 7 cells that appear at different stages of development, some appearing embryonically, some appearing post-embryonically. And then finally the C, which shows kind of the placement of these cells in time. So you have hypodermal 7 cells forming in the embryo, and then you get the larvae. So you get these post-embryonic hypodermal 7 cells, and then they fuse and then contribute to structures in the adult. So those are kind of some of the things, and I have it written up a little bit better than that. I'm not going to pull up that source document, but just to show some examples of what kind of how that proceeds, that process proceeds. There's also, of course, there are these nuclear divisions which weren't represented in there, where you get once the cells fuse, you get nuclear divisions, and they contribute to this number of the several rounds of nuclear division depending on the structure, and that's why we get those larger numbers of nuclei. Do you have any questions or comments?
I just wonder how it all fits in with the mechanics.
Yeah, well, especially when you get these structures where you get fusion of cells. And I'm going to talk about mechanics next, actually.
Oh.
Yeah, I've got some interesting things on that. But yeah, it's like, when the cells are fusing and they're forming these structures, they're kind of coming from different places. So there's some migration and, they're coming together in a certain way. So the migration aspect is something that, that's where you'd have the spatial dimension, right? You have like these lineages. I showed them kind of in sort of a lineage tree mode, but like they're not spatially correct. They're not kind of in space and then plugging in. So if you had like a spatially correct lineage tree, and I don't mean like in terms of anterior to posterior, I mean like a three-dimensional spatial representation. They would come together in certain ways. There'd be a mechanical there'd be mechanical aspects to that sort of to migration and then the fusion.
Which cells are touching another cell and which ones are deformed?
Right. Yeah.
Like sort of the forces that come to bear. Like if you're getting a fold, you'll have different pressure than if it's flat.
Lalith said, these visualizations are quite informative. I was able to comprehend both the lineage and sublineage level information, both fission and fusion directions. Yeah, well, I guess it is fission when you're talking about cells dividing, and then fusion when they're coming, I don't know if they're coming back together, but there's this cell fusion process going on. So it's, but it's, yeah, definitely like that sort of reticulation in the lineage tree. Something we haven't talked about a lot in this is that, there is this reticulation of the cell lineage tree, where, it's not just that it's branching constantly. That's the whole point. There's some coming back together of that.
Okay, Bradley, were those figures ones you generated?
Yeah, I drew those out, and that was just based on like the lineage tree and then some information from the literature.
Okay, are they fully compatible with that table I showed you?
Yeah, they should be, except for the number of nuclei, which isn't represented there. It's just like, the cells, which have one nucleus per cell until you get to the fused cells, which then isn't specified.
Okay, so maybe we could continue that effort and get to a point where we've got a satisfactory articulated lineage tree.
Yeah.
Okay, which shows all the information we have.
Yeah.
Okay, that's terrific. Thank you.
Welcome. Yeah. Yeah. So yeah, it's just interesting what you can do with like the available data out there. C. elegans was a good model organism for that. Okay. So in promising Susan some mechanics here, I have a paper and then so I want to talk about this paper here, which is this new paper, Emergence of Cellular Nematic Order is a conserved feature of gastrulation in animal embryos. So this is about gastrulation, but it's also about nematic or cellular nematic order. And so what does that mean, nematic order? And so that's where we have to talk a little bit about physics simulations and about soft active materials and about things like liquid crystals. So, this is where we have, cells being like these oblong things or these circular things that have this collective organization in terms of their orientation to one another. So in tissues, a lot of times cells will be oriented in different ways. That might be aligned with each other, that might overlap with each other. There might be some like orthogonality to their arrangement, so they're organized maybe in parallel, organized perpendicular in a perpendicular manner, and all that. This is a software package called Freud, which has nothing to do with psychology or anything. This is a Python library to analyze trajectories obtained from molecular dynamics or Monte Carlo simulations. So a lot of times when people do physics simulations, they'll do molecular dynamics simulations, and those give them the tools to do like things like simulating the physics of cells. So there are a lot of molecular dynamics functions that are available. People do a lot of things with like, you know, particle physics and with, biochemistry, where they have to work out the molecular dynamics of something. And then of course you use Monte Carlo simulations to simulate sort of the stochastic nature of those events. And so this library supports many input formats and outputs NumPy arrays, enabling integration with scientific Python ecosystems for many material science workflows. So they use this a lot in material science. And what we're talking about here is material science in a sense, because we're talking about these kind of soft active materials. And so, what we're talking about are these liquid crystals where we have these rod-shaped or, oblong type objects that are arrayed in some sort of matrix. There's this thing called nematic order, which is where they're aligned, versus a smectic order or some other type of order where they're not aligned in the same way. And so what you want to do is you want to be able to compute this sort of nematic order parameter, which basically allows you to take the orientations of these objects, which in our case are cells. They could be molecules, they could be particles, and determine their order in terms of the orientation. And so, like most complex systems, where we want to know, in a group of objects, what their collective behavior is, we have an order parameter. And so this is the equation we use to calculate the nematic order parameter. So I just took this this is an AI overview, but it's largely from the Freud documentation on Read the Docs, and it kind of goes through some of this. So this is basically the elements of a 3x3 matrix where they're computed, and we take the particle orientation vectors, which have some heading, and then we multiply the components together. We sum overall the particles in the system, so we see it has an integral there, and we basically get this order parameter. So it's basically, it's not really an average, it's kind of like, a collective measurement of all of the orientations. And so this gives us this degree of alignment in this order parameter S. So basically, when we get this order parameter S, we can tell whether things are perfectly aligned or whether they're random. So if they're random, S is near 0, and if they're perfectly aligned, S is near 1, I believe. So that's what we're dealing with. And so that it kind of gives you some breakdown of the formula. Again, the interpretation here is that S of 1 is where we have perfectly aligned molecules where all of the angular measurements equal 0 degrees, and S equals 0 are randomly oriented molecules where the average theta is 54.7 degrees. Again, there's no pattern there. And then an S of negative half or negative 0.5 is where molecules are aligned perpendicular to the director. So there's like one set of molecules that are oriented one direction and another set that are oriented in another direction. And that's actually not random. That is, there's an alignment there. It's just not nematic. And so we can actually get a sense of how these molecules, or in this case cells, are aligned using this simple parameter. And so they say in real nematic liquid crystals, the order parameter typically falls between 0.3 and 0.9, with the exact value depending on factors like temperature. So these are in physical systems. Now in biological systems, they don't know if temperature plays a role here, but this is something that we're going to talk about in the paper. So this is where you have a nematic order parameter of 0.03. So this, you can see that there is no order here. These trajectories are at all different angles in this space, and so it's very much, they're all kind of in different orientations. An order parameter is 0.5, where there's a director, and then there are some other orientations. You can see that there's a little bit more order. They're kind of in the same direction, but they're a little bit, off. You see that you're starting to get the sort of nematic order, but it's not quite there. And then finally, an order parameter of 1.0 almost looks deterministic. It's perfect nematic order where everything is oriented from left to right. You can see every orientation is the same pretty much.
Yeah, Bradley, has this been applied to swarms like birds and fish?
Yes, I believe so. The math is the same. And I don't know the papers off the top of my head, but I've seen some where they've talked about the sort of, where you're looking at the trajectory heading of the individual agents and, basically asking the question, is this ordered or disordered?
Yeah. Okay.
Okay, so then that brings us to the paper of the day here. This is the Nature Communications. It's actually, I guess it was published online this week. So this is John Wallingford is on this, Mark Piefer, they're developmental biologist, Robert Hubner. And so they're basically trying to figure out whether we can look at cells in development and say whether or not these have some sort of nematic order to them. And how does that happen? So the abstract reads, cells undergo dramatic morphological changes during embryogenesis, and how these changes affect the formation of ordered tissues remains elusive. Remember, in this case, we're talking about individual cells sort of forming a tissue, but they're orienting themselves in different ways. So you might be forming like skin cells, or you might be forming heart muscle cells and things like that. In order, the orientation of the cells is important in that sense. Here we show that a phase transition leading to the formation of a nematic liquid crystal state during gastrulation in the development of embryos of fish, frogs, and fruit flies occurs by a common mechanism, despite substantial differences between these evolutionarily distant animals. So basically, the embryos in all these model systems, they looked at them and they found that there was this nematic order that was playing a role here. And remember, this doesn't necessarily have anything to say about mechanics explicitly. There's an implicit sort of discussion about what kinds of mechanics lead up to this nematic order, and what kinds of mechanics does nematic order imply. And so that's I think maybe the question here with respect to mechanics. But this is something that's happening kind of as a way to sort of maybe it's to maintain the integrity of the tissue. Maybe it's for signaling. The cells need to signal one another and kind of be aligned. Maybe they're migrating and they need that nematic order to migrate properly. Importantly, nematic order forms early, before any discernible changes in the shapes of cells. So this nematic order is an early sort of precursor to shape changes in the cells, and by extension, differentiation of the cells. So you can have nematic order before that. All three species exhibit similar propagation of the nematic phase, reminiscent of nucleation and growth mechanisms. So this is where we have this nematic phase grows across the cells, and it propagates. So this ordering isn't just like local; it can become larger over space, I guess. And the spatial correlations in the nematic phase in the notochord region are long-ranged and follow a similar power law decay, which is given here, with alpha less than unity, indicating a common underlying physical mechanism. So this means that a lot of these processes, this process in these different model organisms happen according to they happen according to this power law across a certain anatomical region. So you have local nematic order, and then you can measure, whether if you start at one point, whether that nematic order persists across the region, and that gives you some idea of maybe how it starts and how it propagates. And this is a very similar sort of local versus global relationship in all these different organisms, which suggests that there's some sort of physical mechanism that's very much common to every organism. So that means that there's probably a role for mechanics here, setting up this sort of, setting up nematic order in these different structures. To explain the common physical mechanism, we created a theoretical model that not only explains the experimental observations, but also predicts that the nematic phase should be disrupted upon loss of planar cell polarity, which you see in the frog. You also see cell adhesion in the frog, and the notochord boundary formation in zebrafish. So let's go back to this. This is where they're trying to figure out the common physical mechanism. They created a theoretical model, and they're able, with this theoretical model, to predict that the nematic phase should be disrupted in cases where you lose planar cell polarity, cell adhesion, and notochord boundary formation. Basically disrupting these mechanics and seeing what happens, or these physics of the embryo, and seeing what happens. Gene knockdown or mutational studies confirm the theoretical prediction. So you can actually disrupt all of these processes by knocking down certain genes and disrupting this process. So cell adhesion, they're adhesion molecules. So planar cell polarity, you can disrupt that. And then notochord boundary formation. All those things can be disrupted by some sort of, using either defined mutants or some sort of knockdown strategy. And this confirms what their theoretical model suggests. The combination of experiments and theory provides a unified framework for understanding the potential universal features of metazoan embryogenesis. So this is something that you'll see across animal embryos. And so this is, again, going back to maybe some of the first embryos. What were going on in those first embryos that we have? I think we talked about this a while back, and I did a blog post on the origins of embryos, where they were finding these embryos in paleontological assemblages. Some of them looked like collectives of cells, so they were like multicellular aggregates, and then some of them kind of look more like modern embryos. And so that's worth thinking about. This sort of nematic order, is this something you see very early in embryo evolution? That's something you'd see maybe in these very early multicellular aggregates. And if you go to multicellular aggregates and say something like yeast, would you also see these kind of nematic order transitions, or do they play a role in it? So it's kind of interesting that there's this very deep phylogenetic history of nematic order in embryos. So basically, we want to understand the advent of ordered structures, and this is one way to do this.
So, Bradley?
Yeah.
In looking at the formation of Syncytia, I came across a very interesting case which seems to contradict this work.
Okay.
And that is, some cells actually make a loop and fuse with themselves. And that's not nematic at all. Once they fuse. Okay. So it's zero nematic, and yet it's very strange. It forms tubes. The cell turns itself into a tube. Okay, and it doesn't fit their model. Okay, So it's an interesting exception.
Yeah, yeah.
I mean, I wasn't aware of this until I was looking at various forms of cell fusion, which could or could not lead to syncytia. And obviously, if a cell makes itself into a ring, it still has just one nucleus, and so it's not a syncytium.
Right.
Okay, so I discounted that as an example of a proper syncytium, but it is an exception to their structures.
Yeah.
Okay, so while they think they've got something universal, they obviously don't.
Well, yeah. I mean, I don't know if it's for every structure in the.
Yeah, I know. Well,
Yeah.
they're touting it as.
Yeah. So yeah, this kind of talks about what they see as this evolutionary conserved process of convergent extension, where you go to narrowing and elongating of the body during gastrulation, where you get this transformation from largely spherical early embryos into the elongated animal body plan with this head to tail axial organization. So you get this sort of convergent extension and this is something you'll see across species. And again, this convergent extension is driven by directed movement of cells in one axis, which they call convergence, resulting in extension of the perpendicular axis. So you have convergence along one axis and extension along the other. So it's like going from a kind of a sphere down to this elongated form. That's basically what they're talking about. And then, so this is something that people have sort of studied for the history of embryogenesis, but it remains unclear how cellular scale behaviors translate to larger scale physical transitions. And so this is kind of what they're selling as universal. And if it happens across a lot of species, you could make the argument that it's universal. There are always going to be exceptions, however, and in different systems as well. So it's kind of like when you say something is universal, you're kind of they have to specify the mechanism quite extensively, especially in biology. So confluent cell populations, such as those found in the early embryo, can exist in a variety of physical states. For example, a high-density tissue consisting of isotropic, non-proliferative cells will be in a jammed or solid-like state. So this is interesting in a lot of the sort of the liquid crystal literature, in the soft active materials literature. They talk about these different phase transitions of matter. And one of them is that you have maybe like a free-flowing flow of particles that are moving in a maybe like cells migrating, and they're doing this freely. And then at some density, as they increase in density, they jam or they freeze up and they stop moving together. And so basically the idea is that, if you think about this as a linear process, you have a bunch of particles or cells that are migrating. And if this were a linear process, they would just simply slow down gradually and then freeze at some density that, where you might expect it to freeze. There's no, kind of sharp difference in the behavior of those cells or those particles. In the case of jamming or this jamming phase transition, you get this free-flowing state, this migratory state, and then at some critical parameter value, they jam and they become a solid, and it's quite an instantaneous event at this parameter value. So this is an interesting thing about the role of order parameters and phase transitions in developmental biology as an extension of material science or soft active materials, in that the collective behavior of cells can change quite suddenly and be subject to these phase transitions. So, this is in contrast to a growing tissue undergoing dramatic morphological changes where they must be in a more fluid-like or unjammed state. So you get these unjammed states and jam states, and you get these phase transitions in between. And I think we've run into a number of papers where people have proposed that there are these different transitions between jammed and unjammed states. So sometimes, development will proceed by having like an unjammed state where cells can migrate, and then a jammed state where cells are kind of stuck and in stasis, and then another, it transitions back to an unjammed state where cells then are free to migrate again. And this is all governed by some sort of density. So if you reach a critical value of density, if the cells become dense enough, they freeze up in these states. And so this is what they're kind of getting at in development. And then this kind of leads to the order or the orientation of the cells, because the orientation is important in determining this kind of density, because if they're all in the same direction, they can pack more densely, and if they're not in the same direction, they can't. It also governs, other types of migratory behaviors or other things like that. So this kind of goes through the nematic phase and how this kind of works. So here's some figures. You can see that these cells here are in these different orientations, and they're able to track them and look at their orientation over time. And they have a section here that says the onset of the nematic phase occurs prior to major changes in cellular anisotropy. So anisotropy is where you get this difference in orientation, and you get these major changes in cellular anisotropy, and the nematic phase occurs prior to those changes. So they use the spatiotemporal changes in the alignment of cells to determine the mechanism of formation of the nematic order as convergent extension proceeds. To this end, we first assessed the correlation between the shape index and the nematic order parameter, and they looked at the spatiotemporal dependence of SI and S, which are their parameters. At early times, there are no discernible changes in SI along the medial lateral axis. In contrast, a dome-like shape appears in S along the same axis, indicating the presence of a nematic phase in the medial lateral region. So there's this local nematic phase, and it's governing this sort of transition, suggesting that changes in orientational order precede changes in the cell shape. So again, the order of the population of cells maybe locally can precede how the cell changes its shape. So basically the cells are becoming ordered, and then they're changing their shape, and they're undergoing this kind of change in global phenotype or global morphology. Of interest, microtubules also displayed polarized growth prior to overt cell shape changes, suggesting a role for the microtubule cytoskeleton in early nematic order formation. So much as we know from mechanics, cytoskeleton is playing a large role, and microtubules especially in changing these conformational shapes and changing how cells interact with one another.
Bradley,
Yeah.
I have a question. They say the behavior of the cell precedes its shape.
Yeah.
But determining shape requires an inaccuracy in measurement.
Yeah.
Okay? Could it be that they simply don't have enough spatial resolution?
That's possible. I mean, and again, like, shape is a tough thing to measure in a lot of ways.
Yeah.
Yeah. So yeah, I don't know. That's interesting. I mean, I don't know how I'm not going to get into too much about the measurements, but it's possible. Yeah. And there are a lot of variation. There's a lot of variation in shape that we don't understand the significance of too. So
they need to look at actin ring dynamics.
Yeah. For instance, they seem to contradict themselves in the sense that the microtubules elongation may be the primary thing, and it's got to start at by the addition of single tubulins, which they can't detect, and yet may have an influence.
Well, if you want to get into tensegrity, it's both the microtubules and the actin ring acting together to change the shape.
Yeah, what I'm saying is,
tension
look,
in the cell.
there's got to be some sort of feedback between microtubule extension and cell shape.
Right.
And you could easily miss the beginning of that because the beginning of that process occurs at extremely low resolution, small scale, namely the size of individual tubulins. So I can't agree with them that change in cell shape comes after the process of nematic formation. I think they just didn't detect it.
Yeah.
Okay, I mean it's how should I say it's similar to standing a pencil on its point. Yes, it falls, and you may not be able to predict the direction it falls, but the initial phase of it falling could be detectable by having a sufficiently high resolution picture of the pencil.
Yeah.
Okay, and even though you can't predict it, once it starts, it's determined.
Yeah.
Okay, so
Yeah.
that conclusion of theirs is not convincing.
Yeah. So the last thing I wanted to talk about with respect to this paper is a sort of long-range spatial order. So they talk about this here to characterize the nature of nematic order in the notochord regions. This is a different area, different thing that they're talking about. We calculated the spatial correlation, describing how the alignment of cells varies with distance. So remember, we have these local effects of nematic order, and then we can look at the global effects, but we have to understand like how the nematic order might decay over space. So, we have a local area where there's high nematic order. It might decay across a certain structure or region, and then, what is the sort of consequence of that? So, an exponential decay of the function would indicate that alignment weakens rapidly as the distance between the cells increase. So, as cells become less densely packed or less, um, have less sort of density between them, as they're sort of packed together, you might have a rapidly weakening alignment. So this is all, of course, dynamic. So it's not just enough to say we can take a snapshot of the region or of the cells and say, okay, they're at this orientation, so this is going to happen. There's this active process where the cells are constantly remodeling, the cells are constantly changing their density, they're constantly moving against one another. And so we can only take the averages for any one area, local or global. And so this is another problem that you have with these kind of collective systems, is they're constantly changing. So you might take a static measurement, you get this dynamic effect because there are all these things happening kind of continuously. So anyways, but there is this relationship between density of the cells and nematic order and decay over space. So if there's this exponential decay, that would mean that alignment weakens rapidly. However, if this decreases slowly, exhibiting a power law pattern, and this is different than linear decay, where you just have this linear decay over space, it suggests that there is significant alignment even between cells at large separation, indicating long-range order. So in other words, the order is long range in terms of space. There's this, it ranges over a larger area of space, and it's not so much the local density, it's the global density that's controlling this process. So they did the spatial correlation, which is this parameter C sub SR, this function, and this is how they define it, where SRI is the nematic order of cell at location RI. And the average is over all cells in the notochord region. So they're looking at this global average. Strikingly, the decrease in correlation function follows a power law. And so this is how you state that. With a small value for alpha, indicating the presence of a long-range spatial correlation in the notochord region. The value of the exponent alpha changes in a time-dependent manner, but remains small throughout, which is consistent with the finding that the nematic order in the notochord region emerges early in the convergent extension process and remains persistent. So this is where you get this early emergence and then this persistent effect. Taken together, these data reveal that the formation of the zebrafish notochord is accompanied by the growth and propagation of a nematic phase starting from a seed, reminiscent of a nucleation and growth process observed in crystals. So this is a liquid crystal model. So there's this analogy between the embryo and the liquid crystal and how these nucleation and growth processes occur. Observation of a nematic liquid crystal phase during zebrafish convergent extension prompted us to determine if this liquid crystal state is an evolutionarily conserved feature of convergent extension. And when they say evolutionarily conserved, it could be that there's, it's probably more of a physical parameter that governs a lot of embryonic systems. I think that's probably a good way to put this, because that's, common processes are happening across different model systems. So there's a lot more in this paper. I'm not going to talk about a lot of interesting figures, and of course their measurement techniques, which I'm not going to get into. But I think it raises a number of interesting questions with respect to sort of collective behavior, and the embryo is a complex system, as well as sort of these mechanical issues about how cells behave in a tissue. Did we have any other comments or questions?
Yeah, I think they didn't simulate this behavior at an intracellular level, did they?
I don't think so.
No. Okay. I'd suggest, if somebody wants to tackle it, a simulation of microtubules inside cells and their effect on cell shape change and how they affect whole tissue. Is the notochord a 2D or 3D tissue? Is it an epithelium?
Everything's 3D.
Everything's 3D. Okay, fine. It doesn't matter. What I'm saying is that as the microtubule elongates in the cell, one could try to calculate the effect on the next cell. And there might be some feedback in orientation there, let alone communication of, I don't know, how do you get cell-to-cell communication on tubulin concentration or tubulin condensation rates? But I think there's a simulation to be done here to see what parameters cause the cells to align and whether you get your power law or not. Okay, so
yeah, a lovely power law.
Well,
okay, this is a power. You could, one thing, for example, microtubules have this dynamic instability where they elongate rapidly. Well, no, actually, they elongate slowly, I guess, and then collapse rapidly.
Yeah, it can.
And that kind of thing could definitely influence alignment of neighboring cells, because while it's elongating, it's kind of acting as a stiffening element. When it's shortening, it's less stiffening. Okay?
Yeah.
So I think a simulation of the nematic properties based on intracellular microtubule might be worth looking at. In realistic molecular fluctuations. Microtubules, for instance, can bend. They can be stiffened by lateral interactions. Literally, as we showed by a factor of 10 to the fourth. And there could be all kinds of intracellular things which are having a large influence in setting the initial drive towards the nematic phase of a tissue. Okay, so it might be worth tackling this at a molecular level.
Right.
I wonder if I should be taking that workshop again. It's a whole weekend. It's Python programming, so yeah.
Yeah.
What's the relationship? I don't understand.
Anyway, it's CompuCell 3D. It's that modeling program, and they're having a workshop, and they do interactive. You interact with their program, and then you do a project at the end.
Oh, okay. Oh, I see. It's you're not just learning Python.
No, you're programming in it. So yeah, you're learning it while you go.
Okay. Okay. At any rate, as I said, the start of an instability, such as alignment of nematic structures, might have to do with the statistics of molecules inside those structures. And
yeah. Yes.
all about their growth and their collapse and their bending and their attachments to other things.
And how many of them are there?
And how many are there? Right. How many are there? Their relationship to microtubule organizing centers. So
yeah.
the initial phases of nematic formation of tissues is approachable.
Yeah.
Okay.
Well, I know where microtubules are in cells, and they tend to line the sides and crisscross.
Yeah, the side ones, the ones at the apiglans that are perpendicular to those.
Yeah.
It's Beth Burnside's work. Okay, so yes, they do all sorts of things, but the mechanics, therefore, of an individual cell and its relationship to the tissue might be an approachable problem.
Well, I know it depends on its attachments, and microtubules are involved in that. So its attachments to its base and the attachments to the cells to the side are very important in its mechanics.
Who's attached to the side?
They're attached to the trijunctions.
To the what?
Trijunctions. Tri or quadjunctions.
Junctions would be at the ends.
At the where the cells meet. Where 3 cells meet, or 4 cells meet, or 5 cells meet. The junction.
They actually, microtubules attached to those points.
Yeah.
Oh, that's curious.
Well, I don't know that they go across it, but the microtubules form the base. And then you get linking molecules in between.
Yeah, there's all sorts of literature on bundles of microtubules also, which might be involved in the but I haven't seen any on formation of the bundles.
Oh, okay, yeah. Well, there was something.
So
too much literature.
Too much literature.
Yeah.
It's enormous. Okay, but yeah, I think these nematic properties of tissues could possibly be explained.
Okay, yeah, there's different types of cell sheet movement, apparently.
Yes, but this is a chicken and egg problem they presented us with, and they've claimed that the chicken comes before the egg.
Yeah. Okay.
Okay. So I kind of doubt that.
Well, anyways, this
I mean, any process which explodes in the sense that it gets bigger and bigger requires a starting point. And it might be a small fluctuation, but it does require it.
yeah, you need a. So what starts that?
I mean, yeah, right, exactly. What starts it? Are fluctuations sufficient, or do you have to add something? Like stiff microtubules with lateral connections might do it.
Okay, well,
So,
I haven't studied that part of it because I was just doing stationary to ignore the cell sheet movements
oh
stuff.
yeah, I know, but
Working on it.
I know
Yeah.
that's hard enough, but
But yeah, they've said, oh, there's 3 different types of cell sheet movement now. Hello. There's a new type of cell sheet movement out there. Yeah, just recently published
Really?
paper. Yeah.
Okay,
Yeah,
should
well-
send that along here.
Now, if I could find that, but if you could do cell sheet movement and then a new form of it, maybe
Yeah, I
you
was
could
surprised.
find it.
I was surprised in this stuff about cell-cell fusions. I thought there were only a couple of them, and I ended up with a list of 6 different kinds of cell-cell fusions.
Yeah.
Oh, okay.
Okay,
Yeah.
and that includes this ring one, which contradicts this whole business about pneumatic order.
I was also reading something that said, yes, cells sort of have a pneumatic order, but it's not quite the same as a crystal. Like,
No,
Right,
they're
right.
sort
Yeah.
of
It's
critiquing that.
it's kind of a metaphor that's applied because it's not exactly the same material.
Yeah, I know.
Right.
It's a universal metaphor, which is not universal.
Well, I guess that's all for today. Thanks for attending. All right.
All right.
See you next week. Thanks.
Okay, bye.
Bye.
Bye.